The ability to direct cell-cell interactions has proven valuable in a variety of applications, including adoptive immunotherapy, tissue engineering, and regenerative medicine. One approach is to introduce an engineered receptor onto a cell's surface, enabling that cell to target an antigen of interest. Perhaps the most popular and successful example of this is the chimeric antigen receptor, or CAR. In this approach, T cells are genetically engineered to express a new receptor that targets a surface marker on neoplastic cells. While the genetic engineering approach is successful, it does have some limitations. For instance, the permanence of the expressed receptor leads to severe side effects, and this has so far restricted the use of CAR T cells to hematologic malignancies. Furthermore, not all cell types, such as stem cells, are amenable to the genetic engineering needed to introduce the receptor. Finally, the engineering process is tedious, inefficient, and relatively expensive. To circumvent some of these limitations and potentially expand the use of membrane-engineered cells, our group has developed a non-genetic approach to reversibly modify cell membranes with targeting elements. The scaffold we use to do this is called the chemically self-assembled nanoring, or CSAN. The CSAN is comprised of two components. First, a targeting element is fused to 2 units of the enzyme dihydrofolate reductase. These monomeric fusion proteins can then be spontaneously oligomerized into nanorings by a chemical dimerizer, which we call bis-methotrexate, because it's essentially 2 units of the DHFR inhibitor methotrexate that have been chemically linked. Importantly, the CSAN can be disassembled through exposure to the antibiotic trimethoprim. Trimethoprim is a stronger inhibitor of DHFR, thus it outcompetes the methotrexate for the active site and drives the disassembly of the CSAN. Using this system, we hypothesized that CSANs could be installed on cell surfaces and used to direct reversible intercellular interactions. To accomplish this, we needed to address 2 initial questions. First, what should the targeting element be? We wanted something that would be easily engineered into our DHFR-based fusion proteins and something that could be rapidly and solubly produced. This essentially excluded antibody-based scaffolds, such as single-chain variable fragments, and as I've alluded to, we settled on fibronectin domains for this purpose. The other question, which I'll address in the latter half of the talk, is how do we attach the CSANs to a variety of cell types? Specifically, we wanted an attachment system that would be relatively innocuous with respect to the modified cell itself, and something that could be directly applied to multiple cell types. As I mentioned, we decided to use a targeting scaffold based upon the human 10th type 3 fibronectin domain. As shown, this domain is comprised of 7 anti-parallel beta sheets, all connected by flexible loop regions. 3 of these loops, depicted in red, form a congruous surface on one face of the protein, and it's been extensively demonstrated that the amino acid sequences of these loop regions can be mutated and evolved to form a binding paratope for a target of interest. Furthermore, these evolved ligands have demonstrated useful properties, including small size, thermal stability, and a lack of cysteines, and thus disulfide bonds. The target we were interested in is called epithelial cell adhesion molecule, or EpCAM. EpCAM is an established cancer biomarker that's overexpressed on a number of carcinomas, and it's also been shown to be expressed on cancer stem cells. So the question then became, how do we transform a wild-type fibronectin into a high-affinity and selective binder for EpCAM? For those that aren't familiar, we used a directed evolution methodology to accomplish this. Briefly, a naive library of randomized fibronectin sequences was created at the DNA level and then transformed into yeast for surface display. We then sorted this expressed library against EpCAM in a variety of contexts, which I'll discuss. After each round of phenotypic selection, the remaining clones were diversified largely through loop shuffling via homologous recombination and random mutagenesis with error-prone PCR. After repeating the cycle a number of times, we were ultimately able to isolate fibronectin clones exhibiting high affinity and selectivity for EpCAM, which I'll show you. Now, like many others, we started by sorting the library against recombinantly produced biatenylated EpCAM that we adhered to magnetic beads. Though we isolated clones that bound very well to the recombinant antigen, we found that they bound poorly to EpCAM expressed on cells. We felt the issue is that there must be some fundamental structural difference between the recombinantly produced EpCAM that we'd been sorting against and the way EpCAM is expressed and displayed by mammalian cells. Therefore, we turned to mammalian cell panning as our selection approach in an effort to isolate only those clones that could bind to cellular EpCAM. We took an early sublibrary that had been partially enriched through the recombinant antigen sorting, and we panned that against adherent monolayers of EpCAM expressing cells. We used two different cell lines for this, MCF7 and LN-CAP, and we panned against the EpCAM negative cell line U87 as a control. After several rounds of panning, we identified clones that bound selectively to the EpCAM positive cells, as demonstrated by these phase contrast microscopy images. The small spherical cells are the yeast binding to the plaque-like MCF7 cells, whereas no detectable binding is observed on the U87 cells. Unfortunately, when we titrated the affinities of these clones against the MCF7 cells, we discovered that they were largely in the high nanomolar to low micromolar range. We felt the issue now was that we had panned yeast expressing numerous copies of the fibronectins against cells overexpressing EpCAM. Due to the high avidity between the yeast and the mammalian cells, the affinity of an individual clone did not have to be very high to survive our selections. What we needed was an approach to isolate high-affinity clones from our cell panning endeavors. At the time, however, no reproducible method for affinity discrimination existed strictly for cell panning. Fortunately, our collaborators in the Hackle lab were developing a system to address this. They noted that the displayed ligands are tethered to the yeast surface through a pair of disulfide bonds. They were able to show that by treating the yeast with the reducing agent dithiothreatol, or DTT, that they could reduce a proportion of the disulfides and lower the valency of the displayed fibronectins. Using a model system of previously reported EGFR binding domains, they were able to show that by reducing the valency of the yeast-displayed ligands, they could selectively enrich a high-affinity binder from a population of medium and low-affinity clones. For instance, the 2 nanomolar clone, shown in black, starts at about 1% of the initial mixture. When sorted without DTT, we see some enrichment of this clone, but the effect is much more pronounced when the yeast valency is reduced to between 6,000 to 3,000 ligands per cell. In fact, the desired 2 nanomolar clone becomes the most predominant one in the mixture under the right conditions. If you reduce the valency too much, however, you hamper the ability of any of the yeast to bind to the monolayer, and you return essentially the starting mixture. Based upon these results, we sought to apply this avidity reduction approach to our EpCAM selections. To test whether the approach would work outside of the model system, we panned the population under both reduced avidity and standard conditions in parallel. After several rounds of panning, we expressed a polyclonal mixture of ligands from each population and assessed their binding to EpCAM-positive cells by flow cytometry. The histogram plot here shows the control in black, the binding of an early, relatively unmatured population in red, the binding of the population sorted under standard panning conditions in blue, and the binding of the population sorted with the DTT treatment in green. As shown, the avidity reduction approach yielded a population comprised of higher affinity binders than were obtained through the standard sorting methods. To characterize this effect further, we then sequenced, expressed, and purified a number of individual clones from each of the 3 populations. As indicated by the representative size exclusion chromatograph, the clonal ligands exist as monomers. We also titrated the affinity of each clone against cellular EpCAM using the MCF7 cells, and the binding curve for one of the high affinity clones is shown here, with a KD of about 17 nanomolar. After characterizing a number of clones, we found that those isolated under the reduced avidity conditions had a significantly higher affinity than those isolated under the standard approach. The range of KD values for the high affinity clones was about 11 to 30 nanomolar. Furthermore, the clones remained robustly selective for cellular EpCAM, with essentially no observable binding to the EpCAM negative U87 cells via flow cytometry. So having developed these EpCAM binding fibronectin domains, we then sought to incorporate them into our Csans. We fused them to our DHFR scaffold as outlined here, and after a few rounds of optimizing the necessary linker lengths and identities, we generated species that are solubly expressed and rapidly purified. They also maintain their binding efficacy and selectivity for EpCAM. In this case, we did experience a 2-fold loss in affinity from 17 to about 35 nanomolar, but we still observed no off-target binding, here using EpCAM-negative Raji cells as the control. Furthermore, the individual fusion proteins are purified as monomers, and in the presence of the bis-methotrexate dimerizer, they readily and completely oligomerize into Csans. We can further demonstrate nanoring formation by cryo-TEM, where we observe the Csans themselves. Our size analysis indicates they have a mean diameter of about 19 nanometers, and this correlates well to dynamic light scattering measurements we've obtained for these species as well. So having developed the targeting ligand and incorporated it into our Csans, We now sought to install them on cell surfaces, again with attention to our criteria. We took advantage of the fact that we can further derivatize that bis-methotrexate dimerizer. Specifically, we can synthesize any third arm that terminates in a primary amine. We can then use this amine to conjugate a variety of cargo. For this project, we were initially interested in conjugating phospholipids, as these might enable the C-sands to hydrophobically insert themselves into the cell membrane. However, these lipid-based constructs proved to be incredibly difficult to synthesize and purify, so we began to think of other ways we may be able to achieve a similar outcome. For that, we conjugated a bioorthogonal click tag to the amine, specifically a small peg with a terminal azide group. We then envisioned that we could incubate cells with commercially available phospholipid conjugates, such as a disteroyl phosphoethanolamine linked to a dibenzocyclooctine group. The phospholipids would hydrophobically insert into the cell membrane and display the DBCO groups. We could then click a preformed C-SAN onto the cell surface using a copper-free ligation between the azide groups and the strained alkyne, ultimately granting EpCAM targeting capabilities to the modified cell. We also envisioned a similar approach using biotinylated phospholipids. Rather than a click reaction, we fused a monovalent streptavidin domain to the DHFR subunits, thus forming bispecific C-SANs that could interface between the biotin-modified cells and the EpCAM-positive targets. After finding that the hydrophobic insertion of these phospholipid conjugates has no effect on cell viability or proliferation, we demonstrated that we can detect the presence of the biotin and DBCO moieties on the cell surface by probing for them via flow cytometry. Notably, the lipid modification can be performed in two ways: by resuspending the cells in a phospholipid-containing buffer, or by simply growing the cells in culture media supplemented with the lipid. The modifications are also tunable in the sense that more or less of the biotin and DBCO can be installed on the surface simply by varying the concentration of the lipid used. The biotin approach is seemingly more efficient, likely due to the fact that the biotin-streptavidin interaction develops much more quickly than the slower click ligation between the DBCO and azide. Finally, this method was successful for all of the cell types tested, and I've shown data for adherent MCF7 cells and suspensive Raji cells as examples. We then demonstrated that we could install a variety of C-SAN species onto the lipid-modified cell surfaces. Specifically, we can bind streptavidin-based C-SANs to biotin-modified cells, we can bind azide-based C-SANs to DBCO-modified cells, and we can bind EpCAM-targeted C-SANs to EpCAM-expressing cells. Furthermore, we can form bispecific C-SANs functionalized with both the monovalent streptavidin and fibronectin domains, and we can bind them to both biotin-modified and EpCAM-expressing cells, demonstrating their bifunctionality. Now, because these are lipid-based modifications, we were worried that the lipids may rapidly dissociate from the cell surface, as they've been shown to do in other contexts. While we do see significant loss of the individual lipids over the course of 3 days, the lipid-anchored C-SANs remain stably bound to the cell surface, likely due to the multivalency afforded by the C-SAN. That is, each nanoring is tethered to multiple lipids and not just one, giving it an enhanced avidity for the cell surface and facilitating this stability. We also wanted to demonstrate that we could still remove the targeting elements from the cell surface by disassembling the nanorings with trimethoprim. When C-SAN labeled cells were incubated with clinically relevant concentrations of trimethoprim at 37 degrees, we observed a time-dependent loss of the targeted subunits from the cell surface, with 95% of the modification removed within 2 hours. Finally, we can use this platform to direct and reverse cellular interactions. For the fluorescence microscopy experiment shown in the top row, CFSE-labeled Raji cells were modified with the biotin lipids and functionalized with the EpCAM-targeted C-SANs. They were able to associate with a monolayer of adherent MCF7 cells even after extensive washing. That interaction was then readily reversed with trimethoprim. A similar result was observed by flow cytometry, where the functionalized Raji cells were able to interact with the MCF7 cells, forming targeted cell clusters that survived the high flow conditions of the cytometer. Again, the intercellular interactions were reversible with trimethoprim. Putting it all together, we've developed a cell surface engineering approach where a self-assembled nanoring scaffold is installed on a lipid-modified cell surface. The C-SAN can then direct targeted cell-cell interactions, which are readily reversed with trimethoprim. To conclude, I've shown you how we evolved the first EpCAM-binding fibronectin domains, and how mammalian cell panning under reduced avidity conditions was essential for the isolation of high-affinity clones. We then incorporated those ligands into our CSANs, which were installed on cells that had been hydrophobically modified with phospholipid conjugates. The CSANs were then able to direct intercellular interactions that were pharmacologically reversible in vitro.